Hello, everyone, and welcome to our COVID-19 virtual town hall. This evening, in collaboration with NYU Health and the LGBT Center, we would like to welcome you. I know it's snowing outside, and this is the kind of weather that you just want to run home and cuddle and watch some nice long movies and drink hot chocolate or hot cider. But we are glad that you're able to join us so that we can talk a little bit more about COVID-19, but more so about the vaccination that we are all hearing about. And what we are doing this evening is that in collaboration with NYU Langone, we've got with us Dr. Mark Mulligan and Juanita Herb from NYU Langone, and they will talk with us this evening about the vaccine, about NYU Langone's role in the vaccines that we've been hearing all about, and what does that mean for our communities. One of the things that is particularly important about this vaccine that I hope we'll be able to address or talk about is, what does this vaccine mean for our communities, especially LGBT people of color communities who because of our race have been historically distrustful of medicine, of vaccines. But let us hear what Dr. Mulligan and what Juanita are likely to tell us about these vaccines and what this vaccine means for our health and us being able to get back to, as soon as possible, to a semblance of normalcy. But before we move on, I'd just like to welcome and introduce Antonio Roberto, who is with the LGBT Center. Antonio, welcome.
Thank you so much, Antoine, and thank you for hosting this evening. Welcome, everyone. I'm Antonio Roberto Jr. I'm the Senior Director of Behavioral Health at the LGBTQ Community Center here in New York City. We're the nation's first LGBT center. We were founded in 1983. We provide a whole array of social services. Since March, all virtual and remote, mostly via Zoom. We provide everything from a youth drop-in Monday through Friday to HIV testing to the only LGBTQ specific substance use outpatient clinics in New York State. Our mission is to empower and help our LGBT community members lead healthy, happy lives. And we are very thankful to have NYU Langone Health here tonight, and thank our collaborators, Encore Community Services, Trans Latinx Network, and Voces Latinas for joining us in presenting this important event. I'll pass it back to you, Antoine. Thank you again.
And thank you very much, Antonio and the LGBT Center for hosting this, because I think it is crucial that this conversation is happening in conjunction with the LGBT Center so that our LGBT community members can have a sense of what this vaccine means for them. So to begin with, I would like us to take a moment to spend a moment in silence as we consider those in our communities who have died from COVID-19 or who have died from suicide or homicide in our communities. Thank you very much. Now, usually, because of these online virtual forum and so that we're doing these days, there are a couple of things that we need to keep in mind, and we call them community agreements. So we ask that you don't take any screen. We ask you to be respectful of everyone who's participating in this screen. And if there's a chat box, you can put questions in that you want to put questions in. So the general question, and let's just last question. At the end slides. So the entire this event is being recorded and will be available at the LGBT Center. So our first presenter is Dr. Mark Mulligan. He's a clinical trial lead and principal researcher. He will be talking about the safety of COVID-19 vaccine trial overview. Of the Division of Infectious Diseases and Immunology Department of Medicine at the NYU Vaccine Center. He joined NYU on 2018. He's a translational scientist who leads a research clinic and research laboratory. He conducts vaccine clinical trials and clinical studies of emerging infections. He has studied HIV, Zika, Ebola, bird flu, the 2009 pandemic influenza, and other infections that has a public health impact. He is now studying the novel coronavirus. Dr. Mulligan is a graduate of the University of Notre Dame, cum laude in 1980, the University of Texas Southwestern Medical School in Dallas, 1994, and during infectious diseases training at UAB in 1987 to 1990, he did postdoctoral molecular biology training with Dr. Richard Compands, a renowned virologist. Dr. Mulligan served as lead investigator for NIH project HIV/AIDS clinical trials with conducting AIDS and key research in Atlanta, Kenya, the Philippines, and Canada, which is from 2016 to 2018. He also served as principal investigator for an NIH project vaccine and treatment from 2007 to 2018. He was one of 9 national units funded. This is one of 9 national units funded by NIH.
Antoine, I just want to cut in for a moment. Your audio has been coming in and out. So I just wanted to pause for just a moment and see if there's any issue you can detect on your end before we continue. Can you test right now?
Hang on, maybe I need to stop sharing.
You're sounding okay now.
Okay. So let's pick up. So Dr. Mulligan was participated in one of 9 national units funded by NIH to conduct clinical research on medical countermeasures, such as vaccines, treatments, diagnostics, and biomarkers to combat infectious disease threats to human health. Community members, I'd like us to welcome Dr. Mark Mulligan. Dr. Mulligan.
Thank you, Antoine, for that very gracious introduction. Thank you to Antonio. It's a real pleasure to be here for me. I look forward to this evening talking a bit about the vaccine and continuing with the discussion as possible. And I'm particularly happy that my clinical operations manager, Nurse Juanita Herb, is joining us tonight. And I'll just say that this is a community that is so important to all of us and one that I've worked in for many, many years. And I look forward to working with you, I hope, for many years going forward. I'd like this to be the beginning of a relationship where we can continue to support one another. So with that, I will go to screen sharing. And Antoine, is my screen looking okay? Antoine,
yes.
or Antonio, someone?
Yes, Dr.
Yes,
Mulligan.
it looks okay, Dr. Mulligan.
Okay, great, thank you. All right. Well, let's go ahead and get started. This is such an important topic. First of all, we are an NIH-funded center for vaccines and treatments designed to fight against infectious diseases. And tonight we're going to talk about COVID-19. And It's actually very interesting. I'm sure that many of you are familiar with the NIH-funded HIV clinical trials network, such as the ACTG, the HVTN, HPTN, and I've been an investigator in all of these. Well, COVID-19 is of such national import that NIH has basically turned all of its clinical trials networks, including all of the HIV/AIDS clinical trials networks, to fight against COVID. And of course, we must continue the fight against HIV/AIDS and try to come up with a cure, a vaccine, even easier PrEP, etc. But for now, we also, as Antoine very rightly said, want to get back to a closer version of our normal life. And in all likelihood, a COVID vaccine will help us do that. And so that's what this talk tonight is about. At the NYU Langone Vaccine Center, which I direct, our mission is to protect and restore human health through infectious disease clinical trials. And we're interested in working in all communities, in children, in older adults, and younger adults. And we can't do our work without community. Community is essential. If you think about it, everything in our medicine cabinet, all those prescription medicines, they are there because of clinical trials and clinical trials study participants, communities, individuals who really helped in learning about the safety and the effectiveness of all these medicines. And so community is absolutely essential, particularly with vaccines where there's sometimes hesitancy, and particularly now in the current climate where there has been reason for people to lose faith and lose trust. We also must work to regain trust, particularly in our communities of color and from underrepresented minority communities. And it's a time of fear, of uncertainty, isolation, mistrust, and misinformation. And tonight, what I hope to do, what I'd like to do, is to offer a chance to fight for the future, some optimism. We will be science-based tonight, fact-based. Like to make the point that together we can defeat COVID-19. My hope is that we can end COVID-19 in 2021. And we will try to address some of the misinformation and mistrust that has been out there. Well, let's go ahead and get started. So what is a vaccine? How does a vaccine work? So a vaccine trains our body to recognize a virus like SARS-CoV-2, the COVID-19 virus, should we come in contact with that virus after getting vaccinated. So the vaccine trains our immune system. And then if we do get exposed to the virus at some future time point through our daily lives, maybe going to the grocery store and someone nearby us coughs, for example. And even though we have our masks on, masks are not 100%. They are effective and we should wear our masks. But despite that, as you know, we're seeing a lot of COVID, in some cases because people aren't wearing their masks. Although I think in general here in New York, we're doing well. Not entirely, but compared to many parts of the country, we're doing well. In any event, if that virus then tries to come into our body after vaccination, the trained immune system trained by the vaccine sounds an alarm, and immune cells like lymphocytes and antibodies fight against the virus and try to eliminate it so that we don't get sick. So that's really what a vaccine does. It trains our immune system for some unknown time point when we might encounter the virus so that we can fight it off and not get sick. This slide shows several different vaccine platforms or types of vaccines. There are many different candidates that have been studied and are being studied. There are the RNA vaccines. There are weakened viral vectors like recombinant adenovirus, and then several other classical vaccines like protein vaccines, sort of like a flu shot or others. I'd like to spend a minute on the RNA vaccines. So there's been a lot of media attention to the Pfizer-BioNTech vaccine. We'll just call it the Pfizer vaccine. This is an RNA lipid nanoparticle. In essence, it's a copy of the gene encoded in RNA for the spike protein of the virus. If this is SARS-CoV-2, the COVID-19 virus, that red structure there is the spike protein. If you're familiar with HIV, this is equivalent in essence to GP120 or GP160, which is on the surface of the virus. In the case of HIV, that protein binds to the viral receptor for HIV, CD4. In the case of COVID-19 or SARS-CoV-2 virus, the spike protein binds to the receptor for SARS-CoV-2 in lung cells, the ACE2 molecule. And so it's very important protein for the SARS-CoV-2 or the COVID virus in terms of entering our body, entering cells, and making us sick. So if we can block that, it'll be very important. And so the RNA vaccines have the genetic code for the spike protein. And when these are injected into the muscle of our arm, we give an intramuscular injection into the deltoid arm, the big muscle at the top of your arm, just the same place we give the flu shot. The RNA vaccine goes into our muscle cells, and then the way I'd like you to think of it is the RNA is like a message to the cell, and it tells it, make the spike protein. Not the whole virus, because the whole virus isn't there. It's not encoded by the RNA, just one aspect, one component. And the body makes the RNA or the spike protein, the vaccine spike protein, and then our body makes an immune response, the T cells, the B cells, antibodies. So that's how an RNA vaccine works. The RNA is a message. You can think of it as an email or a text message if you want to the cell. The cell then makes the vaccine spike protein, and the body then makes the immune response. So 1, 2, 3, RNA message to the cell, make the protein, make the immune response that on future encounter would protect us. And I'll mention right now that the Pfizer RNA vaccine and also the Moderna RNA vaccine have been incredibly protective, 95% protection in very large clinical trials, phase 3 trials enrolling 75,000 participants combined. So really good news with these RNA vaccines. Currently, we're enrolling in, and we've participated in the Pfizer vaccine trial at NYU. From the very first day that a human being received the Pfizer vaccine, we have been vaccinating at NYU since May 4th and following up the participants, studying the safety, the immune response, and the protection levels. In addition, we're now enrolling and vaccinating with the AstraZeneca vaccine candidate. And this is a non-replicating or non-multiplying viral vector. It's a harmless common cold virus. It can't make humans sick. And it encodes the spike protein as well, just like the RNA of Pfizer or Moderna. Moderna and Pfizer RNA vaccines are very similar. In the AstraZeneca vaccine, the initial encoding is as DNA within this adenovirus vector or carrier. And the DNA then leads to production of RNA, then you get the protein, then you get the immune response. So it's just starting one step earlier. Instead of RNA protein immune response 1, 2, 3, as with the RNA Pfizer vaccine or the Moderna RNA vaccine, with the AstraZeneca adenovirus vector vaccine, it's 4 steps: DNA, make RNA, make protein, make the protective immune response. And we know from phase 3 trials in Brazil and in UK that the AstraZeneca vaccine has, in one study, 64% protection, and in another study, 90% protection, an average of 70% protection in those 2 overseas phase 3 trials. We're now doing a very large NIH-funded trial of this vaccine, and we're enrolling right here in New York City. And I'll just mention that we have 5 clinics across New York City, vaccine research clinics, 3 in Midtown Manhattan, 1 at the Tisch NYU Hospital, 1 at Bellevue, the City Hospital, 1 at the VA. We also have a vaccine research clinic at our NYU Brooklyn Hospital and also at our NYU Long Island Hospital in Mineola. All 5 are participating in these clinical trials of COVID vaccines. And there are other candidate vaccines. This one, as I mentioned briefly, is a protein vaccine. So you're not delivering DNA or RNA, you're just delivering the protein, and then the body makes the immune response. So that's a simpler kind of vaccine. And in fact, most of the flu vaccines are these protein type vaccines. The Sanofi and the Novavax are 2 companies whose make protein COVID-19 vaccines. And we will be participating probably in February in a Sanofi COVID-19 vaccine. So far at NYU, we've participated in an RNA vaccine, Pfizer. The Moderna vaccine was studied at many other centers around the world. Participating currently in AstraZeneca, recombinant adenovirus vector vaccine, and we will be participating in the Sanofi. Important unifying point across all of these, they all present the spike protein to the immune system. So that's a critical protein for neutralizing antibodies and also a target for T-cell responses. I want to spend a minute on what it's like to develop a vaccine. So how is a vaccine developed, approved, manufactured? Well, after the vaccine has made it through preclinical testing, which usually involves animal testing, laboratory testing, FDA will review the proposed first-in-human study, which usually involves 20 to 100 participants. And then the trial can begin if FDA gives their approval. They feel the design of the study is appropriate. And this study has one major question: is the vaccine safe? Are there any side effects? And then you start to learn about the immune response, the antibodies and T cells. If the vaccine is safe in phase 1 and produces an immune response that we believe might be protective so that it's worthy to proceed to phase 2, we then enroll, rather than tens of subjects, a few hundreds of subjects, and we continue to learn about safety, and we learn more about the immune response. If the vaccine still appears safe and producing an immune response that we still believe would be protective in phase 2, we go to phase 3. This is the final phase of clinical testing before a request for licensure. And in phase 3, we're asking, does the vaccine protect? We've already established that it's safe and that it produces an immune response that we think would be protective. And now we ask, does it protect? How do we find that out? Well, it's a randomized trial. Half of the participants get vaccine, half get a placebo, a saltwater shot. And it's blinded. The participants don't know which they're getting. And I, as the investigator, Juanita, as our nurse, we don't know what the participants are getting. And then we follow. We never intentionally expose our trial participants to COVID-19, but rather through their daily lives, people in the middle of a pandemic, an epidemic, are going to come in contact, unfortunately, with the virus. And so we have the chance to determine if receiving the vaccine results in fewer infections occurring, fewer illnesses occurring, compared to those who got the placebo or the saltwater shot, the dummy vaccine. So If a vaccine looks effective in phase 3, then the manufacturer can go to the FDA and apply for licensure. In the case of the COVID-19 pandemic, because we're in a public health emergency, FDA is providing an early form of approval called an EUA, which you've heard about in the news, emergency use authorization. It's not the same thing as full approval or licensure. The vaccine is not yet fully approved, but the FDA decides based on all the evidence and based on a public meeting, and this happened last week for Pfizer, that the totality of the science indicates that the known benefits outweigh the known risks of using the vaccine for prevention of COVID-19 disease, in this case, in people aged 16 years and up. So we now have our first COVID-19 vaccine.
It's 7 o'clock.
Excuse me, that was my computer talking. We now have our first COVID-19 vaccine with an emergency use authorization. And in fact, this week, as you may have seen on the local news, on Monday, we started vaccinating at several hospitals around New York City. I'll say more about the vaccines in just a minute. But to continue with the clinical trials, sometimes after a vaccine is improved, there will be what are called phase 4, some studies to continue to learn even after the vaccine is approved. We have national systems, including this one, VAERS, for reporting of side effects even after a vaccine is approved. Because sometimes a side effect can be so rare that it may only be 1 in a million, and you won't be possible to learn about it in a clinical trial of even 100,000 people. So here's an important question. Can the COVID-19 vaccine cause infection or illness? Can you catch COVID-19 from the COVID-19 vaccine? And the answer is a very emphatic no. The vaccines are made in a laboratory. They never contain the whole virus, so you can't catch an infection or the disease from the vaccine. One way to think of it is if this bicycle is the whole virus, what you would have to get exposed to in order to cause COVID-19 disease, the vaccine is just one piece of it. Let's say the front wheel. And it's never the whole bicycle. So here we're showing the spike protein or some RNA, but never the whole COVID-19 virus. So very important point. In clinical trials, or now that we have a vaccine available for use in the community because of the EUA that was issued last week for the Pfizer vaccine, and we expect an EUA for the Moderna vaccine, another RNA vaccine, later this week. It's being reviewed at FDA by their expert external panel tomorrow. Now that we have the vaccine available, important to know that you can't catch COVID-19 from these vaccines. So when someone participates in a clinical trial that is looking at safety and effectiveness or efficacy or protection, what happens? So before, during, and after, we always answer all questions. There's a written informed consent document that someone would sign in order to give their approval to be in a study. You can drop out of the study anytime you want. You don't have to continue, and there are no hard feelings about that. But once you've had a chance to consider being in the trial, we schedule you for a first visit. And that's where we do the informed consent process. There'll be some blood collected before vaccination, then the vaccine is given. And usually there are 7 to 9 visits during the clinical trial over 2 years for these vaccines. And for the Pfizer vaccine, there are 2 vaccinations, 21 days apart. So the people that got their first dose at NYU are doctors and nurses and janitorial staff and medical care techs in the hospital, frontline medical workers. They're getting vaccinated this week at NYU, and many hospitals around the city. They will come back in 3 weeks and get their second dose, the booster dose. So it's a 2-dose regimen, this vaccine. And then after vaccination and even after the study, we still continue to follow up with people. The studies last 2 years, so we can learn all about the safety. So what does it mean when we say the Pfizer vaccine is 90% effective? What that is saying is that in the clinical trials of 45,000 people for the Pfizer vaccine, half got vaccine, half got placebo. There were 170 people who became infected with COVID-19 through their daily lives, community exposure, maybe family exposure, maybe exposure at work. Of those 170 infections in the Pfizer vaccine trial, which we participated in at NYU, 162 of the infections were in the placebo group and only 8 in the vaccine group. And the groups were equal size. Half of the participants got placebo, half of the 45,000 got vaccine. 162 versus 8. And when you do the mathematics on that to determine the level of protection, it was a 95% reduction in infection and actually in laboratory confirmed COVID-19 disease, not just infection, but disease in the vaccine group. The other point I'd like to make is that these RNA vaccines are highly effective at preventing against severe disease. For example, in the Moderna trial, the other RNA vaccine, which will be reviewed at FDA tomorrow, there were 30 severe infections in the trial. And when the Data Safety Monitoring Board cracked the code to see where those severe infections were, placebo group versus the vaccine group, do you know how many were in the vaccine group? 0. All 30 were in the placebo group. So there were no severe infections in people who had received the vaccine. Well, where are we now? And I'm moving to the end of my comments. Where we are now is really a time when we have hope. A vaccine is now being rolled out. But I think it's really important for me to say that we have to manage expectations a bit, because it won't be available for everyone because the supply is so limited initially. Eventually, everyone will be able to get the vaccine that would like to get it. What the US government, the FDA, the CDC have recommended to the state, and what the state is requiring of us as a hospital, is that we start by vaccinating residents of long-term care facilities, so nursing homes and other long-term care facilities, and healthcare providers, those who are on the front lines taking care of our patients. Soon, probably in January, February, there'll be more vaccine available over time. Every week, there'll be a bit more available, and we'll be able to start vaccinating those who are older adults that we know are at very high risk for COVID-19 disease and death. Other essential workers, so mass transit workers, bus drivers, grocery store workers, people that have to work out in the community to make their livelihood and are critical to keep our society functioning. Those people would be among the higher priority groups. People with chronic medical conditions, but it'll slowly unroll over time based on the recommendations of the CDC, the FDA, and the state as we get more and more vaccine available. I mentioned already that we're currently participating in the AstraZeneca vaccine. This is the recombinant adenovirus vaccine. It's the phase 3 trial, so we're asking, does the vaccine protect? All 5 of our vaccine center research clinics are participating in the trial. So the 3 in Manhattan, Mineola, Long Island, and Sunset Park, Brooklyn. And we're particularly looking to enroll in this trial individuals that are at higher risk for severe COVID or for acquisition of COVID. So again, people with chronic medical conditions, those who may get exposed at their job, and people over the age of 65. And we have this registry. This is our website, and there's a registry there. If anyone is interested, they're able to go there and provide their information, and we will follow up about possible, well, answering questions, doing some education, and allowing you to learn more about this possibility, this opportunity. Remember, we won't be able to defeat COVID until we achieve herd immunity. We think that's going to mean about 75% of our communities, of our population, vaccinated and protected against COVID-19. It probably won't be achievable till the middle of next year. But hopefully, if the vaccine supply continues to increase, and if there is uptake amongst community members, we can achieve herd immunity and end COVID-19 in 2021. This is how CDC has set the goals for recommending who should be in the first groups to get the limited supply of vaccine, trying to decrease death and serious disease, preserve society functioning, reducing those who are already burdened heavily due to disparities, inequities, and trying to increase the chance that everyone can have health and well-being. But absolutely essential here will be education and rebuilding trust in science, in clinical trials, in vaccines, in government. I truly believe that we're entering a brighter new era where we can rebuild trust. And I must say that the fact that these vaccines are so effective, 95% protective, I hope will help encourage people. And the vaccines are very, very safe. They have excellent safety profile in these large trials enrolling thousands and thousands of participants. And now that we have vaccines that either are available for use in, beginning to be used in our hospitals, I think it's important to say that the vaccines themselves don't save lives. I mean, they're sitting there in the vial. What we need is to have communities and individuals that are engaged, educated, and willing to take the vaccines. Otherwise, the vaccine does no good if we don't get it into people's arms. And smallpox is an example of the power of vaccines. This was a horrible disease. 100 years ago, a significant percentage of children died of smallpox. The mortality rate with smallpox was 10 or 20%. New York City had an isolation hospital out on Roosevelt Island in the East River, and you can see the ruin of it to this day. And happily, it is a ruin because, why? Because smallpox is eliminated due to the smallpox vaccine, which was another highly effective vaccine that was used in the first half of the 1900s and all the way up until about 1980. And we no longer even vaccinate children against smallpox because it's been eradicated from the face of the earth. So that's an example of what we hope to have happening with COVID-19. So I'll just close by saying it really is a privilege for me, and I know Juanita feels the same way, to do this work. And we really hope that we can partner with you in defeating the COVID-19 pandemic. So thank you very much. Thanks again to Antoine and Antonio. And with that, I am going to stop sharing my screen and turn it back over to you, Antoine.
Thank you very much again, Dr. Mulligan. And before we go on to the panel section and to speak with Juanita, I have some questions that were coming up in my mind as I listened to your presentation. And I think, so tell me, what makes these vaccines different? Why are there different pharmaceutical companies doing supposedly the same vaccine? Why is this?
Yes.
Second question is, the Moderna vaccine is supposedly able to be stored at 4 degrees Celsius. as compared to the Pfizer vaccine, which has to be like minus, you know, what is it, 40 degrees Celsius.
Yeah.
Why is there such a difference in terms of temperature controls? Because one of the things that we've been seeing is that for many facilities and hospitals, it's extraordinarily increased cost to put in refrigeration or freezing capabilities to get to minus 40 degrees Celsius to store these vaccines, which only have a room temperature shelf life of about 2 hours. So why is there such a disparity? So you wanna jump in on that, to
Sure.
begin with?
Yes. Let me answer the first question. So we need multiple winning, safe, effective vaccines. If you think about the population of the Earth, it's 7.8 billion. In the US, we have 330 million. We're only 7% of the world's population in the US. We need vaccines for the whole world. We need vaccines for the whole country, for all those that want to take the vaccine. And I hope that will be most people, so we can achieve herd immunity. In order to do that, Antoine, we need multiple manufacturers to be producing safe and effective vaccines. Even if they're somewhat similar, like Pfizer and Moderna, they are independently developed RNA vaccines, and that's totally fine. It's just like flu shots. There are half a dozen companies at least that make our seasonal flu shots, and that's important to ensure a supply. That way, if for some reason there's a problem at the factory of one, the others can pick up the slack. So I think that's my answer to the first question. On the second question, absolutely very important. The storage conditions of the Pfizer vaccine require that ultra cold freezer, and that is not available in smaller clinics, smaller hospitals. That is a logistical problem. It is available at NYU and many major academic and large hospitals in the city. The Moderna vaccine, the other RNA vaccine, can be in a regular freezer, like the same temperature as our kitchen freezers. The best of all in terms of this cold chain logistics is the AstraZeneca vaccine, which can be just in 4 degrees, which is the common refrigerator temperature. So they do vary. What I would say is, because we're in this deadly pandemic and we have in our country 3,000 deaths every day, that those logistics are going to be addressed head on to the best that we can. What we'd like to do is get to maybe down the road new formulations of these vaccines that might be more friendly in terms of the cold chain requirements. But for now, that's what we have. We're glad to have them, but it is something that we would like to see improvement. So it's a great point.
Thank you very much again, Dr. Mulligan. So I'd like to bring in and introduce Juanita.
Hello.
Juanita Herb is she is a Black queer femme who currently serves as the clinical research operations and nurse manager at the Vaccine Center. In addition to her research work, Juanita writes and speaks about intersectional LGBTQ+ identity on platforms such as The Advocate, Huffington Post, South by Southwest, and prior to the pandemic, she produced some of the largest LGBTQ fashion, beauty, and identity events in the United States at reputable institutions such as the Brooklyn Museum and the California Academy of Sciences. She currently serves on advisory boards for NYC Pride and NYC's LGBTQ Film Festival New Fest, and is a contributing editor at Curve, the nation's best-selling lesbian magazine. So community members, I'd like us to welcome Juanita. Juanita, welcome.
Thank you. Thank you for having me.
So in this section of our presentation, we'll be talking about participant experience in the clinical trial process during COVID-19 research. But before I get to that, and I think my question will be for both you and Dr. Mulligan. So we learned about the COVID virus, about the end of January, beginning of February. And it began to take hold significantly in our communities in March, late February into March. And then everything started going crazy. But we also know, and I've heard from a lot of people, that they have been so deathly ill from the end of September, October, November, and parts of December. And they were experiencing some of the same symptoms. We know that there is a recorded instance of a 56-year-old woman in San Francisco who was diagnosed post-mortem with COVID because she had contracted COVID back in November or December last year, but it was not known that she hadn't traveled anywhere. So my question is, when we think about this virus, how were we able to get to produce the antibodies for it in such a short space of time? Because considering, and in our prepared questions, you saw I had a question about HIV, and I'm going to come back to that at another time, little later on. But I want us to talk about how were we able to bioengineer the vaccine so quickly in less than 10 months. Who wants to jump in on this one?
Juanita, do you want to start and then I'll follow up?
Well, sure. From what I understand of the way that vaccines are developed is it's usually based on the body's natural ability to clear vaccines. So HIV and COVID-19 are different, but I'll let Dr. Mulligan talk a little bit about the mRNA vaccines, which are a little different.
Sure. Yeah, you're absolutely right. And one of the things about HIV that's different is that it is a retrovirus. It integrates a DNA copy of its RNA genome into our body's genome. And once it's in, unfortunately, it's there to stay. We do have some hope for cure, novel technologies like CRISPR that might be able to go in and cut out that integrated retrovirus. And let's continue to hope for that. On the other hand, COVID-19, although it is also an RNA virus like HIV, it's not a retrovirus and it doesn't infect us for life once we're infected. And in general, those kinds of viruses are easier candidates to develop vaccines against. So I think that's one reason that although the first vaccine for HIV in a clinical trial goes back to 1987, 33 years. I did my first HIV vaccine trial in 1994, 26 years ago. And sadly, we don't yet have an HIV vaccine. It's not for want of trying. It's been a very difficult, wily opponent, unfortunately. Antoine, for COVID-19 also, it's benefited because all of the science that has advanced for decades, much of which has come from work on HIV. I think that's one beautiful thing about science is that you can learn from your neighbor and sort of beg, borrow, and steal from other conditions to work on your particular condition. So when this pandemic started, there was the RNA vaccine platform that had been developed over several years and was being evaluated for cancer vaccines, for HIV vaccines, for CMV, Zika. And really it was a matter of quickly adapting the RNA platform to insert that spike RNA from COVID-19 virus and then proceeding with the clinical research. So we were able to move in months, Antoine, even though it's been 33 years and we still don't have an HIV vaccine. We were able to move in months against COVID-19 because we had a pre-existing platform, because it was a virus that maybe in a way we got lucky. It's one that we could effectively combat with a vaccine. It's not a retrovirus. It doesn't integrate into the body's DNA. And frankly, a lot of hard work and effort put forth around the world to move these candidate vaccines forward. So I think that it was the timing of it was right. And I think people are asking, well, gee, if these RNA vaccines work that well for COVID-19, maybe now they could be adapted and evolved to work against HIV, a better TB vaccine, tuberculosis, maybe a better malaria vaccine. So it is going to be an interesting time. But I think as a pandemic response platform that's rapid and adaptable and flexible, easy to produce lots of doses, this will be a wonderful tool for the next epidemic or pandemic that we need a rapid vaccine for. You can bet that they're going to apply the RNA platform.
Thank you very much, Dr. Mulligan. And I think I am glad that this forum is being recorded, because you just said something just now that I think will be able to put, or should help, hopefully be able to put a number of people at minds at rest, especially those who are living and struggling with HIV, because there's been a significant amount of anger and frustration in many communities that are people of LGBT people who are living with HIV, who are saying, well, why is it an HIV vaccine can't be produced after 40 years, but we can get a vaccine for a virus in less than 10 months. So I think that what you just shared is important, and hopefully that there is significant funding and investment from both private and government to invest in gene editing and CRISPR that can work on the HIV virus in a similar way as they were able to do with the COVID virus.
Right.
Juanita, because this virus went through such a short production time, there was not so much an ability to undergo the complete clinical trial process. Okay? What about a phase 3 clinical trial that needs to be completed? And if it wasn't completed in entirety, what can be done while we are where we are with this emergency use authorization? And I remember Dr. Mulligan saying just now that examinations and research will be continued another 2 years. But what happens in that phase 3 trial that people should be aware of, that people should know, and to be prepared for?
Yes, absolutely. So currently our Pfizer phase 3 participants are still in the clinical trials, and we will follow them for 2 years. In addition to our AstraZeneca phase 3 trials. Now the AstraZeneca phase 3 trial, the AstraZeneca platform has not received the emergency use authorization, but those who are enrolled in both of those clinical trials are followed for 2 years. Now I know that there was some confusion in one of our last town halls where people said, why is it that the Pfizer is done and that the AstraZeneca lasts for 2 years? So both of those trials last for 2 years, and we follow subjects to see. Another question that has not yet been answered is that it is effective, but how long is it effective? Will the antibodies last? So those are some of the outstanding questions that we have. But what we do know from the initial results of the Pfizer trial is that it does provide protection, and that we need to get people vaccinated right away. So people who are receiving it under the emergency use authorization who are not part of our clinical trials will receive it. We also saw, you mentioned in previous conversation that we had offline about Nurse Lindsay, who is the first nurse to get it, first person to get it under the EUA outside of the clinical trials. But our clinical trial participants are going to continue to be followed. Many of them are going to be unblinded, and they will receive the vaccine in the same rollout as recommended by the CDC.
Thank you. One of the things that is also coming up is, for many in our community, this is why I am so insistent on focusing a little bit more on HIV, because many of our Black and people of color, LGBT community members are living with or struggling with HIV. And there is some concern about taking this COVID virus and its interaction with HIV medications that people are currently taking, or with the virus, with the HIV virus itself. So can you or Dr. Mulligan talk to us about this?
Yeah, I'd be happy to. I'm very pleased to be able to say that the Pfizer vaccine trial, the AstraZeneca trial, we are able to enroll people with HIV infection, and they have been included in the trials. And also the Moderna trial did that. Now, these are people that are on medications, stable, well controlled as far as their viral load. The clinical trial has to enroll in these initial trials a group that has an immune system that's relatively intact. Eventually, we'll study all people, even those who have immunosuppression, maybe from advanced HIV, cancer, medications they take for autoimmune conditions like lupus or rheumatoid arthritis. But in these phase 3 trials that have been done thus far, there was inclusion of people with chronic stable HIV infection. And what we learned was the vaccine worked just as well in them. People with chronic medical conditions, including HIV, had the same level of safety and the same level of protection from the vaccine. Even though it's an RNA vaccine and people probably know that HIV is an RNA virus, I don't think there's any kind of interaction between this this virus, the SARS-CoV-2 or the COVID-19 virus vaccine and HIV. Some of you might remember that lopinavir ritonavir, an HIV med, was for a while touted as a possible treatment for COVID-19, and that didn't pan out. The clinical research to date suggested it didn't have activity against the coronavirus, although it is quite active against HIV. So 2 different RNA viruses. I would like to reassure all of our community members with HIV infection that if they have chronic stable HIV, there's absolutely no problem. And if their HIV is a little less controlled, there's not an exclusion. There's not a safety issue. No one's saying they couldn't get the vaccine. It would just be a matter of, I think, risk and benefit. Whenever we lack evidence, which we don't have the evidence yet for people with less controlled HIV, less well-controlled HIV, we don't have as much evidence or data as we want yet. Eventually we'll have that. But when we're in that situation and we're in a pandemic, what is being said, and I believe this is the right approach, Antoine, is that it might be right for you to take the vaccine. If you're, let's say, a postal worker and you're out in the community all the time getting exposed, it's all about the potential for benefit. So that the vaccine might really benefit. So let's say someone's a bartender. Well, I guess now the bars are closed, but a grocery store worker. You know, it's such a safe vaccine thus far. I think the risk of harm is pretty low. So I think it is something that can be considered for all folks who have HIV infection.
Thank you, Dr. Mulligan. And let me just follow up a little bit on that, because I remember when Pfizer announced, when Pfizer and BioNTech announced the vaccine, there was a report by the chairman of Pfizer where he said that there is no guarantee that the virus, even after you've got the vaccine, will protect you from contracting the virus again. Can you talk a little bit about that? And because people are still somewhat concerned about, well, what then is the point of me taking the vaccine if I'm still going to get the virus again?
Yes.
The other question that comes up, and I see Juanita had dropped off and I see she's back, and Juanita, you can probably talk about this, is what should people be concerned about taking the vaccine, about getting the vaccine? What kind of side effects should they expect? And one of the most important things that I've seen and heard is people asking, well, if I get the vaccine and then 21 days later I get the second shot, how long does the vaccine last in my system before I may have to get it again or take another COVID test? Or like I just asked Dr. Mulligan about the Pfizer chairman's statement about no guarantee of contracting virus again. So that just kind of leaves all these windows open. So Dr. Mulligan, do you want to start with that one first, and then
Yes.
Juanita can come in on the longevity of the vaccine?
Sure. Yeah, I'll let Juanita comment about what we've learned about the expected vaccine reactions that any vaccine might cause, and how that works with these vaccines. I'll talk about reinfection. So I think it could be, Antoine, that what you're talking about is the fact that we now know that having had COVID-19 once doesn't necessarily protect you from getting it a second time. It seems to be uncommon or rare, but it can occur. There are a few well-documented cases where when someone had a second illness, they had the nasal swab test, they showed the virus was there, and it wasn't just persisting from their first infection. It was months later, and when they sequenced the virus, they showed it was a new strain. So reinfection can occur. I'd say it's occasional or rare. Natural infection does give immunity for a few months and maybe longer. We're still learning. With the vaccine, even people that have had COVID before were protected in the Pfizer trial. We didn't exclude people by testing them for prior infection. We asked them if they had prior infection. If they did, they couldn't be in the study. But we know that we enrolled, no doubt, many people that had prior COVID-19 in the vaccine trial. And the reason that was done is because when we roll out the vaccines in mass vaccination campaigns in coming months, it may be that 10 or 15% of New Yorkers have already had it, maybe higher. And we need to be sure that the vaccine will be safe for all of those people who will get vaccinated. So in the clinical trial, we wanted to be sure to include those with prior infection, although more unknown to them. We did exclude those who had known infection. And remember, CDC has said that up to 40% of COVID-19 infections may be without symptoms. In any event, when the data were evaluated, Antoine, the vaccine provided protection to people even if they had had prior COVID-19. So I think that there's something about these vaccines, they're so protective that they can actually improve upon that natural immunity that comes after natural infection. And that's why I think it's a very reasonable thing, even if you've already had COVID, to get the vaccine. And in fact, that's what CDC is saying, that they should not be excluded. They should be allowed, if they want it, to take the vaccine. You might say in your first 2-3 months after your COVID infection, not to take the vaccine because you're likely to have a strong immune response then. But subsequent to that, certainly makes sense. And there's nothing wrong with getting it even a few weeks after your infection. No need to get it while you're still sick, certainly.
Thank you, Dr. Mulligan. Juanita?
Yeah, and to speak to what we have seen in terms of the some of the side effects, they've been very mild and expected of what you would experience with maybe a flu vaccine. So there might be some tenderness or soreness at the injection site, maybe some fever, chills, but they're mild and they're short-lived. Some people, for example, I'm somebody who gets my flu shot every year, and I always experience some form of, a little fatigue, a little mild, fever that can be broken with, some over-the-counter Tylenol, and it's very similar to that. What you should expect.
Thank you. Now, I'm thinking, and you mentioned this at the very, somewhat towards the beginning of your presentation, Dr. Mulligan, about as many people, 75% or more of the population should be vaccinated to create a sense of herd immunity. herd immunity as a phrase has become significantly politicized and has become significantly stigmatizing. Can you talk to us a little bit more about what is herd immunity and how does this vaccine help us get there? So what I'm asking you to do is to positify, to make herd immunity a little more positive.
Yes.
So we can understand its function in infectious disease treatment, virology, that kind of stuff.
Yes, thank you very much. Excellent points. When I talk about herd immunity, and by the way, some of my colleagues say we should probably call it community immunity, since we humans live in communities, not herds. But when I talk about herd immunity, I'm talking about vaccine-induced herd immunity. There have been, unfortunately, some proponents who have said, well, maybe we should just let this virus rampage through society. And then, because it's not that bad, and most young people do pretty well with it, and then we will maybe achieve herd immunity. And I'm here to say that that is a horrible idea, that the virus is very deadly. 3,000 deaths a day, particularly for older adults, for those with chronic conditions. It can cause damage to pregnant women and fetuses. So not a good idea. We don't want to achieve herd immunity by letting the virus run out of control. That idea is a non-starter. There even was a country that had that to try as its national policy, Sweden, and they are now awash in COVID-19, unfortunately. So that has been disproven. It's a bad idea. All experts are opposed to that. But vaccine-induced herd immunity, and here's where I'll try to positivify it, if that was your word. Vaccine-induced herd immunity is a good idea. That's what we want. Remember, the vaccine is a harmless way to train your body's immune system to fight off an infection. So let's train our immune systems in at least 75% of us, and that's when we can stop the pandemic.
Yeah.
Thank you.
If I can add to that,
Yes.
you know, I heard the other day too that we want herd immunity through protection, not infection.
Yes.
That's what we want to achieve, and it's particularly important to me that we achieve it through this route because Black, Indigenous, people of color, Latin and Hispanic folks have been disproportionately impacted. The way we've been talking about this, though, is not through necessarily an intersectional lens, and the way in which LGBTQ folks also fall into those groups, and how our LGBT communities already are at a disadvantage in terms of access to housing and employment and healthcare. When there's increased numbers of homelessness and housing and accessibility for LGBTQ people, it also makes it particularly difficult to follow public health measures such as social distancing and sheltering in place. So it's really important to me that we overcome this hesitancy and that we start to have these conversations that we're having with organizations such as the center, which has been such an important part of my experience here, living 20 something years as an out queer person in New York City, because it is heartbreaking to see the communities, my communities to which I belong, dying and suffering in such great rates. You know, we talk a lot about the numbers of deaths, and that's very real. We don't also talk a lot about the families that are left behind, the number of people who are left with hospital bills that are bankrupting them, the number of people who have lost their jobs, and it's been shown that not only Black and Brown people are disproportionately losing their jobs, but that LGBTQ people living at the intersection of those identities have been hit the hardest economically. So we want to not just get back to a sense of normalcy, we need an immediate response to this, and then not return to normalcy in the sense that we need to together combat the structural inequities that led us here in the first place.
Thank you
And
for sharing. I'm sorry, go ahead.
oh, I was just going to say that, I'm a proud partner with Dr. Mulligan in making sure that our clinic has a long-term commitment to this.
Thank you. And I think I have a couple of things that just jumped up when you were talking just now, Juanita, that because if you crawled inside my head, you'd see all the synapses firing. Don't make any comments, Dr. Mulligan. But watch this. And I'm interested that you talk about intersectionality and identities and so forth, because I'm actually doing a webinar in partnership with the New York State Office of Mental Health and New York City Department of Health on January 6th, where I'm talking about inequities and racism in mental health, which then leads me to another question I think either you or Dr. Mulligan may be able to address is, to the best of your knowledge, were LGBT people of color and LGBT community members, by extension, included in trials? And do you know of LGBT, either generally or of color, who were involved in the actual design and production of the vaccine? And the reason I ask that, especially in the latter question, is perhaps just like Lindsay and the doctor from NIH, if there were LGBT people of color or LGBT community members who were part of that process, that they could be made public so that people can see that just as equally they are members of, just like you and me, who were part of this. So can you, I don't know if you're familiar or you're able to answer that question.
Juanita, do you want to say something, and then I'm happy to follow on?
Yeah, to be perfectly honest, I am not aware. We've been kind of hyper focused on our team of researchers at NYU, and even when I heard about Dr. Corbett on the Moderna trial, which we did not work on, and I heard on the news, I sent it to Dr. Mulligan right away because it was so, I lit up like a Christmas tree. I was dancing in my living room to see another Black woman at the helm of this research. And I think that we should be put out in the forefront more, because I think a lot of people, communities of color and LGBT people, Q people, will trust this more, knowing that, their community members are behind this research. I will say, though, that something that was fascinating, and this shows the kind of insidious nature of structural racism, is that I'm very active on social media through my other work outside of research. And when people started posting about Dr. Corbett and Nurse Sandra Lindsay, a lot of people of color were very skeptical about why it was that we were just seeing people now of color being brought to the forefront of this conversation after the EUA. And I think that's something that we need to continue to talk about, is increasing opportunities for visibility for people of color, for queer people in science and medicine.
Dr. Mulligan?
Well, I want to say that just as Juanita got your synapses firing, Antoine, a minute ago, she also made me feel very proud and fortunate to have her as a member of our team. Thank you, Juanita, for those wonderful comments. I do know that we have gay and lesbian participants and gay and lesbian participants of color in our vaccine trial. When I interview them and do their histories and physicals, we often will include a sexual history. And so we become aware of some of these things. And I know that that's the case. We certainly, across the Pfizer and the Moderna trials, had what I think is better than And I like what Juanita said about not just going back to the old normal. Let's make it a better normal. The old clinical trials, we've not done as well as we needed to have done with minority participation. And in the Pfizer and Moderna, overall, they did quite well. They were actually, the sponsors, the NIH, was actually slowing down the Moderna trial to be sure that they increased targeted recruitment, participation, engagement, education, and then offering participation to communities of color. 30 to 40% are Black, African American, Hispanic in those trials. So that's better than we usually do, and that's a good thing. And obviously, for the reason, one of the reasons that we're here tonight is that these communities have been overrepresented amongst COVID deaths and disease. They didn't, I have to say, Antoine, there wasn't a specific targeted outreach that I'm aware of to gay and lesbian groups by NIH. But remember I said at the beginning that the whole HIV/AIDS clinical trials apparatus of the NIH networks, ACTG, HVTN, HPTN, are working on COVID now. And so all of those staff members that are normally working in gay and lesbian communities and trans communities are now working on COVID. So I do have the feeling that there's a welcoming approach amongst our staff for these communities. And I think in reaching out to communities of color, hopefully we are achieving that as well.
Thank you. And I think it's important that you just mentioned that because, and I'm glad to hear that as part of your screening process for participants in the trials, that there were some Black and people of color LGBT community members. Because one of the sharpest criticisms against HIV medications is that Black and people of color were deliberately excluded from the bioengineering for HIV medications in the early days, in the design as well as in the trials. And so to see and hear that Black and people of color LGBT are included in trials for a vaccine like this, which will have impacts on HIV, people with HIV, that is really comforting and really a step in the right direction or progress, if you were. Juanita, is there anything that you might want to share with us about the community aspect of this virus, and particularly about the vaccine impact on our communities, particularly LGBT communities?
Yes, absolutely. So one thing that I have been really driving home with our community engagement strategies is that underrepresented communities are not monoliths, and often many of us exist at the intersection of multiple identities. So for example, when we talk about the LGBT community, sometimes that's often talked outside of the fact that LGBTQ people are every race, religion, national origin, nationality, every background, every ethnicity that you could think of. And so what's happening in our communities, as I had previously mentioned, is that some of those inequities are putting LGBTQ folks at higher risk, particularly when they are also Black and Indigenous people of color and Hispanics. And so the other thing that is a big issue for our communities is that many of our communities, and I was talking with the center members about this, is that many members of our communities are in industries such as sex work, or they're in industries of entertainment and art, and they cannot go back to work without this vaccine. And not having a means of earning, not having a roof over your head, also puts you at risk for a whole host of other diseases and a whole host of other medical problems. And so we really need this vaccine. We really need to, what's hard about it, what's happened historically is those things that you were talking about, is the lack of response to the HIV/AIDS epidemic, the lack of inclusion in research, is that now there's this natural mistrust, even for something that we so desperately need. And so our job is to build that trust and to get that to our communities. So one, serious disease and illness that are direct result of COVID-19 can be reduced, and we can achieve that herd immunity, that positive herd immunity, through prevention instead of infection. But we can also get people back to work. We can also do so in a safe way. A lot of people are hurting in our communities right now. I've seen our communities are impacted in ways because when I talk about homelessness, people are having to go back into the closet or go into places, go back to homes that are abusive and violent. Many of our community resources, mental health resources, are safe spaces that the center, for example, usually has for us in person. Those are all shut down. People cannot get to the clinic to get gender affirming care. They can't get hormones, they can't get clean needles, they can't get HIV treatments. So, we're not talking just about COVID-19, we're talking about a whole host of issues that is negatively impacting our communities. And my work daily is not just to operationalize the clinical trials and make sure that this gets done, but is to work with the communities and work with our team to make sure that we're actually building the trust so that people will take this vaccine.
Thank you, Juanita. And so I just want to go to our questions now. I don't know if you guys can see my Q&A thing, but so I want to go to the questions now, and we've got a couple of questions here. And I think I want to throw this one back at you, Juanita. Approximately how long will the vaccine offer protection?
Yeah, we talked about that earlier in the phase 3 trials. They're 2 years we follow. And right now, that's a question that we can't answer. Right now, we know that it offers protection, but that's something that we're going to continue to study. And the great thing about being in a clinical trial is that we do follow you. You have to enter diaries, and we follow every single symptom, every little fever, every little sore arm that you might have. You have clinicians at your disposal to call, and with your concerns, you get tested regularly. We draw blood to see if the antibodies are still there. So we're following you for 2 full years. And we also do compensate you for your time when participating in the clinical trials.
Thank you, Juanita. Dr. Mulligan. One of the listeners, participants is asking, I'm wondering if you're able to explain why the 5% in the vaccine group still get COVID-19.
Yeah, we don't know the answer to that right now. What I would say in general terms is that these are grand slam home run vaccines, if you will. They're as good as our best vaccines. For example, the HPV vaccine, the human papillomavirus, that's a vaccine against cancer, prevents anal cancers in gay men, prevents cervical cancers in women, prevents head and neck cancers in male or female, and it's 95% protective. It's one of our best. The new shingles vaccine for ages 50 and older is also in the 90s in terms of protection. So this is a great level of protection. We will know the answer to the listener's question eventually, why it is that those 5% didn't get protected. Those studies will be done. We will look for, did they have a difference in their immune response? Was there something different about their HLA, the immunogenetic systems that we all carry to help us fight cancers and infections? So eventually we'll know the answer to that, and it may help us design even better vaccines in the future, although, gosh, it's going to be hard to do a lot better than 95%.
Thank you. So I have another question from another participant, and they're saying, I'm wondering if Pfizer and Moderna are so effective, why is it important to continue to work on AstraZeneca and other vaccines? I know you answered this in terms of a global perspective because it's 7.8 billion people on the planet, but try this again so that they can probably hear your response.
Sure. Yeah, this is something I like to talk about, and it's a great question. You know, we got 2 vaccines, Pfizer and Moderna, 95%. Shouldn't that take care of things? The answer is unfortunately no, because the supply, they will not be able to produce enough of those vaccines for the whole world or even for our country anytime soon. So we need to have multiple winning vaccines. So with the studies with AstraZeneca, with Johnson & Johnson, whose clinical trial, 30,000 participant, is just now fully enrolled. And hopefully in about 3 or 4 months, we'll have the answer, or maybe sooner. One sad side effect, Antoine, of having so much COVID in our country right now is that we can get answers about the vaccines relatively quickly because there's so much viral transmission in terms of running a clinical trial and getting an answer about protection or not. So we need more vaccines in order to get as many people protected as possible.
It's 8 o'clock.
The RNA vaccines will not be enough.
Thank you. And this may be a question for either you, Dr. Mulligan, or for Juanita. Should we continue to wear masks after we're vaccinated? And if so, why?
Juanita?
Yeah, absolutely. We should continue to follow all of the public health suggestions, wearing masks, washing your hands frequently, social distancing if you can. And the reason why is that we do need to achieve that herd immunity, right? As Dr. Mulligan said, the vaccine doesn't work unless it gets into people's arms. So right now, as we're rolling things out, I know people are very excited and also about the vaccine. I also know that they're very fatigued, but we need to continue to practice these public health measures, and we also need to get the vaccine.
And in terms of the why part of that, Antoine, I'll just add that we don't yet know if the vaccines will protect against asymptomatic infection. So if I went and got vaccinated and then stopped wearing my mask, and let's say the vaccines don't protect against asymptomatic infection, remember CDC says up to 40% of infections may be asymptomatic, then I could knowingly transmit to others if I wasn't wearing my mask. Remember, the mask protects me, but it also protects those around me. So that's a really important reason to continue to wear our masks. And I'm saying that we're going to need to just sort of accept that and live with that. And we're just going to have to do it probably until well into 2021, late 2021, assuming we can get to herd immunity.
Thank you, Dr. Mulligan, and thank you, Juanita. But I have one final question. Remember, my training is in journalism, so I always have questions. We see that climate change is beginning to have a significant impact on our world, and by extension, on our very lives. Do you see that this virus may be as a result of climate change? And can we look forward to equally rapid vaccines or treatment for other viruses that we previously had no immunity that may emerge?
Well, I'm happy to take the first crack at that, and then love to hear Juanita's thoughts. I don't really think, I can't draw a line between this virus and climate change. Climate change is real and unfortunate, and we have to change things. One thing that's happened with, it seems like, and it may not be true because it may just be that we didn't have the communications in the old days like we do now to know what's happening all over the world, but it seems like with these outbreaks, we're having more of them in the last 40, 50 years. Zika, Ebola, HIV, COVID-19, bird flu, all of these things. And maybe it is just communications and awareness, and they were always there, and we didn't have a way to know what it was. It was just the fever that was killing people. But it also might be that with global travel and societal changes, that there's more chance for these viruses to spread globally. They may pop up in China, as we believe is possibly true here, and then spread quickly around the world because of jet airplanes. So I think it's more likely to be a result of our international mixing, intercontinental mixing in this case. And yes, I do think that when we have future pandemics, this RNA platform in particular, this rapid response pandemic preparedness platform will serve us well. And whatever that new pandemic virus is, that can quickly be slotted into the RNA and clinical trials can begin. That's how it's supposed to work going forward. And it really worked beautifully this time.
Thank you, Dr. Mulligan. Juanita, you want to take a gander at this one?
Yeah, I mean, I agree with everything Dr. Mulligan had said, and I really do believe that we're doing a lot more traveling, and these pandemics and epidemics can pop up anywhere. As he mentioned, birds flu, we had swine flu. I do think that what will be difficult, though, is responding to pandemics as global warming, as climate change advances, because I think that the way that it will impact the way that we were able to reach communities, and when there are shortages of, possible shortages of food or water, I think that it will become increasingly difficult to get help to where is necessary.
And so thank you very much, Juanita. Thank you very much, Dr. Mulligan. And we would like to, I don't think we have any more questions. Nope, we have no more questions. And so I just want to be able to thank you, Dr. Mulligan. Thank you, Juanita. Thank NYU Langone. I want to thank the LGBT Center, the Trans Latina Network, and Voces Latinas for being part of this this forum. One of the things that I am taking away from this myself is there is now more information about the vaccine. Because one of the things I think that has been lacking in the last several weeks, when we've been talking about all these different types of pharmaceutical companies making the vaccine, was the sense of a lot of information was missing from behind it. So a lot of people did not know. And I think in the absence of information, as you can well imagine, rumor and misinformation and all sorts of falsehoods can run rife. And so once again, thank you very much for being able to be part of providing this information, especially for our LGBTQ communities. Do you want to jump in and you have anything to say, Antonio? Do you want to jump in and say anything?
I want to thank everyone as well. Like I said, our community partners, Trans Latinx Network, Voces Latinas, Encore Community Services. I want to thank Dr. Mulligan and Juanita for being here. Thank you, Antoine, for hosting and moderating, and for the excellent questions and managing of our audience questions as well. And thank you all who are on here listening and whatnot as well. So thank you. I'll pass it back to you, Antoine, to close out.
And so once again, ladies and community members, we want to thank you for joining us for this evening's discussion on COVID-19. I trust that we were able to answer some of your, provide answers to your questions. If you have any more questions or you have any ideas, reach out to the LGBT Center or reach out to Dr. Mark Mulligan or Juanita Herb at NYU Langone. I believe that this recording is going to be shared on the NYU with NYU, and it's going to be on the LGBT Center's website, so you can see and hear some of this information again if you happen to have missed anything, including some of those slides that Dr. Mulligan shared with us. So once again, community members, thank you very much and have a good evening.